In this PowerPoint, we're going to talk about how materials move across the cell membrane, or as we call it, cell transport. So one of the big ideas behind cell transport is this idea of the second law of thermodynamics, which states that the universe tends towards disorder, or an increase in entropy. You may remember this term from chemistry, you may not. But entropy is talking about the amount of disorder. So in general, the universe tends to move towards disorder. If you think about it, if something dies, the body goes from an ordered state to a disordered state of decay over time. So as far as this is concerned with movement of molecules across a membrane, molecules are going to move from an area where they are highly concentrated and more organized to a state where they're more spread out and disorganized. So diffusion is this movement of materials from an area of high concentration. So if you take a look at here, these are orange molecules and they're very packed on one side. So this is pretty ordered. They're nice and structured and pushed close together. So having holes in our membrane will actually drive these molecules to start to spread out until we reach a state called equilibrium. So diffusion is the movement of molecules from an area of high concentration, meaning more molecules, to an area of low concentration. So molecules spread out throughout a space. So molecules move from an area of high concentration to low. So here's dye molecules. They're moving from an area of high concentration to low concentration. I wish that the animation kept going because molecules actually never stop moving. But they move from an area of high concentration to low concentration. And we call this type of movement passive transport, meaning that they're going with the natural flow of things to go from an ordered state to a more disordered state. And to do this kind of transport, no energy is required, meaning no ATP is needed. We can say the same kind of diffusion when we look at movement of water. So here, if you look, this is a cell membrane. We can see the phospholipid bilayer. The green molecules represent solute, meaning things that are dissolved in the water. And then these little guys here are water molecules. And so here, water is going to move from an area of high concentration of water to an area of low concentration of water. And we call this particular type of diffusion osmosis. So the diffusion of water is known as osmosis. So in the last two examples, we saw the molecules move directly through the cell membrane. Sometimes molecules can't move through a cell membrane and they need the help of a protein channel. So channels move specific molecules across the cell membrane, specifically things that are large or charged or polar because they can't fit through the phospholipid membrane. And with facilitated diffusion, again, anything called diffusion is going to require zero energy because we're going from the natural flow of things, an area of high concentration to an area of low concentration. So the term facilitated means with help. So this is diffusion with the help of a protein channel. Active transport is the opposite of passive transport. This is where molecules are transported against the concentration gradient from an area of low concentration to an area of high concentration. And in order to do this, a protein pump is necessary as well as a supply of energy. So the protein pump is going to take molecules from an area of low concentration and move them against the gradient to a high concentration. And this costs energy to do because we're going against the natural flow of the universe. And so it takes effort to cram more molecules into a space that's already full of molecules. And so that energy cost is going to be ATP. Here's another use of our ATP. And to do this again, the energy is actually what causes the protein to have the conformational change or shape change. So there are many mechanisms by which active transport occurs. Down below here we have something called antiport, which is going to transport molecules in opposite directions of each other. So the yellow molecule is going from the left to the right and the red molecule right to left against the concentration gradient from low to high, from low to high. And so therefore it will require an input of energy, ATP. Another model is something called symport, where the molecules are transported in the same direction across the membrane. So both the yellow and the pink molecules are being transported in the same direction. But again, because we're going against the concentration gradient, it's going to require an input of energy, ATP. So getting through the membrane, passive transport, there's simple diffusion, which is diffusion of nonpolar hydrophobic molecules that go directly through the cell membrane from an area of high concentration to low, so things like lipids. And another example of passive transport would be facilitated diffusion. The only difference between simple diffusion and facilitated diffusion is that facilitated diffusion uses a protein channel, so an actual opening in the cell membrane to move the materials across. And again, just like with simple diffusion, we go from an area of high concentration to an area of low concentration. So we're going with the gradient or with the difference. Active transport, however, goes the opposite direction. So this is diffusion against the concentration gradient. Really the pumping of molecules against the concentration gradient from an area where we have a low concentration to high concentration. And in order to do this, it's going to use a protein pump and requires an input of energy in the form of ATP. So transport summary, if we take a look at the top, we have molecules going directly through the membrane, so that would be simple diffusion. Here we have this purple structure here, which is a protein channel. Again, we're going from an area of high concentration to low, so this would be facilitated diffusion. Both simple diffusion and facilitated diffusion are both passive transport. And then finally, the last one would be an example of active transport, where we have molecules moving from an area of low concentration to an area of high concentration against the gradient. And this would require then an input of energy, ATP. So how about large molecules? Everything we've talked about so far have been relatively small. So how would you get something like a large food molecule or a bacterium or something into the cell by bulk transport? So we do this through vesicles and vacuoles. And so we're going to talk about something called endocytosis. So cyto should always mean cell, and endo means into. So taking things into the cell. And we have a couple of different types of endocytosis. We have what's called phagocytosis. Phagocytosis literally means cell eating, and this would bring solid material into the cell. We have what's called pinocytosis, which is cell drinking. Really the only difference between phagocytosis and pinocytosis is phagocytosis brings in solids, pinocytosis brings in liquid. And then the opposite of endocytosis is exocytosis, exo meaning out of, so out of the cell. So endocytosis works by creating little vesicles out of the membrane. So here with phagocytosis, we create little pseudopods, little outcroppings from the cell that engulf the food and pinch inward to form a vesicle. Here, pinocytosis, the big difference is we don't get any of the pseudopods being formed. Instead, the vesicle pinches inward and actually slurps the liquid into a vesicle. And then one we didn't talk about on the previous slide is something called receptor-mediated endocytosis. Sounds really scary, but all it means is that the vesicle won't form until receptors, little proteins, on the surface of the cell membrane are filled with the particular material we're trying to bring in. So what receptor-mediated endocytosis does is it makes sure that the vesicle is full of the desired molecule before it pinches inward and forms a vesicle. So our next PowerPoint will talk about the special case of water, which is called osmosis. So watch that video next. Thanks.